Hello everyone, I am Dr. Deepika Malik. In this session, we'll discuss our stage 3, that is purification. In earlier sessions, we have discussed various methods of cell disruption, stage 1, that is solid liquid separation, and stage 2, that is concentration. Purification is a critical step in downstream processing that focuses on removing impurities and isolating the target product in a highly pure form. It involves a series of techniques and processes to separate, concentrate, and refine the desired product from the crude mixture or solution obtained from upstream processing. The purification step aims to achieve the highest possible purity and quality of the final product. Following are few methods that are used for the purification method. First method are the chromatography techniques that includes adsorption, ion exchange, gel permeation, affinity, high performance liquid, and partition or liquid-liquid chromatography. Then we have membrane processes that includes reverse osmosis and ultrafiltration. Then we have liquid membranes and their types, that is emulsion liquid membrane, supported or immobilized liquid membrane, and bulk liquid membranes. And then finally we have crystallization technique. Now let's discuss these techniques one by one in detail. Coming to chromatography. In many fermentation processes, chromatographic techniques are used to isolate and purify relatively low concentrations of metabolic products. Chromatography usually consists of a mobile phase, which refers to the mixture of substances to be separated, dissolved in a liquid or a gas. As you can see in the diagram, the blue part, that is the solvent, and the orange part, that is the mixture of compounds, when they mix together and they try to enter into the adsorbent or stationary phase, they are termed collectively as the mobile phase. Now, stationary phase is a porous solid matrix through which the sample contained in the mobile phase percolates. As you can see in the diagram, the yellow part. The interactions between the mobile and the stationary phases result in the separation of compounds from the mixture. These interactions include the physicochemical principles such as adsorption, ion exchange, molecular sieving, and affinity. The solution to be injected is usually called a sample, and the individually separated components are called the analytes. So to sum up, the diagram is basically showing you a column in which chromatography is carried out. The column is containing the stationary phase, and the mobile phase, that is the solvent and the sample, enters the stationary phase and gets separated into different components. And the main principle of the separation depends upon the interaction of the analyte and the stationary phase. Following are the different type of chromatographies. Now let's discuss them one by one. Adsorption chromatography. In this technique, the separation is based on difference in adsorption at the surface of the solid stationary medium. The adsorbents such as silica gel, charcoal powder, and calcium hydroxyapatite are packed in the column in a glass tube. This serves as stationary phase. In industries, we might use stainless steel columns as well. Now the sample mixture in a solvent is loaded onto this column. The individual components get differentially adsorbed onto the absorbent. The elution is then carried out by a buffer system or solvent. The most weakly held fraction moves fastest, followed by others according to the order of tightness in adsorption. The individual compounds come out of the column at different rates, which may be separately collected and identified. An automated column chromatography apparatus or fraction collector is frequently used nowadays. So as you can see in the diagram, there is a column which contains the stationary phase of silica or alumina, and when a sample, which is shown green in color, is added onto the column along with the solvent, enters into the stationary phase and gets separated into different components. As you can see, the component Y, that is the yellow one, comes out of the column fast as compared to the component X, that is shown blue in color. So here we can say that the component X is more strongly adsorbed onto the stationary phase, therefore it is coming out of the column at a very slow speed as compared to the component Y that came out of the column at fastest rate. Second type of chromatography is ion exchange chromatography. It is a process that allows the separation of ions and polar molecules based on the charge properties of the molecules. This process can be used for almost any kind of charged molecule, including large proteins, small nucleotides, and amino acids. Ion exchange chromatography retains analyte molecules based on ionic interactions. The stationary phase surface display ionic functional groups that interact with analyte ions of opposite charge. This type of chromatography is further subdivided into cation exchange chromatography that retains positively charged cations because the stationary phase displays a negatively charged functional group, as you can see in the diagram. Here the column is packed with negatively charged stationary phase, and when the sample containing positively charged molecules are added into the column, the analytes basically come out of the column depending upon their positive charge. More the positive charge, more strong is the interaction, and it will take more time to come out of the column. Polystyrene sulfonate resins, CM Cefadex gel, CM cellulose, bear acidic group, and immobilized cations from the solutions. Second subtype is anion exchange chromatography that retains anions using positively charged functional groups. As you can see in the diagram, the column is packed with positively charged stationary group, and when the sample containing the negatively charged ions is added into the column, the least negative molecules will move out of the column at faster rate as compared to the molecules with high negative charge. Diethylaminoethyl cellulose, trimethylaminopolystyrene, DEAE Cefadex bear basic groups and immobilize anions from the solutions. Third type of chromatography is gel filtration or molecular sieve chromatography. Now in this chromatography, the separation of the particles is based on their size, shape, and molecular weight. The apparatus consists of a column packed with sponge-like gel beads, usually cross-linked polysaccharides containing pores. Now this is important. The gels serve as molecular sieves for the separation of smaller and bigger molecules. The solution mixture containing molecules of different sizes is applied to the column and eluted with a buffer. Now this is also important to note that the larger molecules cannot pass through the pores of a gel and therefore move faster, whereas the smaller molecules enter the gel beads and are left behind, which come out slowly. By selecting the gel beads of different porosity, the molecules can be separated. The gel filtration chromatography can be used for an approximate determination of molecular weights. This is done by using a calibrated column with substances of known molecular weights. This is extremely useful in separating ribosomes, viruses, nucleic acids, and proteins depending on their particle size and shape. So here the diagram is showing you a column which is packed with gel beads containing pores as stationary phase. And when the sample is added onto the column, the large molecules prefer to pass through the interstitial space and come out of the column at a much faster rate, since they do not observe any hindrance, whereas the small molecules enter the gel beads and experience resistance, because of which they come out of the column at a very much reduced speed. Next type of chromatography is affinity chromatography. The principle of affinity chromatography is based on the property of specific and non-covalent binding of proteins to other molecules, which are referred to as substrates or cofactors. This technique involves the use of ligands which are covalently attached to an inert and porous matrix in a column. The immobilized ligands acts as molecular hooks to selectively pick up the desired protein, while the remaining proteins pass out through the column. As you can see in the diagram, the column contains the ligands which are attached to the matrix and are specific to particular shape of protein only. Like in this case, only red proteins are able to attach to the ligand, while the other analytes pass out of the column without making any contact. Now this desired protein captured by the ligand can be eluted by using free ligand molecules. Alternatively, some reagents that can break protein-ligand interactions can also be employed for the separation. High-performance liquid chromatography. Since chromatographic techniques are slow and time-consuming, the separation can be greatly improved by applying high pressure in the range of 5,000 to 10,000 pounds per square inch. Hence, this technique is referred to as high-pressure liquid chromatography. It can be applied in any of the earlier discussed chromatography. HPLC requires the use of non-compressible resin materials and strong metal columns. Due to rapidity in action, it is used for assaying amino acids, peptides, proteins, carbohydrates, lipids, nucleic acids, vitamins, hormones, metabolites, and drugs. As you can see in the diagram that the solvent from the solvent reservoir is pumped at high pressure into the HPLC tube that contains the stationary phase. Now this tube is mainly made up of metal only because they have to bear high pressures. In this solvent stream, the sample is also injected. Now the combination of sample and the solvent, which is mobile phase, enters the column and passes through the columns, and the analytes get separated based on whatever principle is being applied, be it adsorption, be it ion exchange chromatography, be it gel filtration or affinity. The analytes coming out of the column are passed to the detector where they are detected. Now coming to partition or liquid-liquid chromatography. The separation is based on the differential partitioning of compounds between a mobile phase and a stationary phase. Now here this is important to note that the stationary phase is typically a liquid coated onto a solid support, such as a column packed with a solid support coated with a liquid phase. The solid support acts as a support matrix to hold the liquid stationary phase in place. The mobile phase is another liquid that is immiscible with the stationary phase. The mobile phase is continuously introduced at the top of the column and flows down through it. It separates solute based on their partitioning between a liquid mobile phase and a liquid stationary phase coated on a solid support. Compounds that have a higher affinity for the stationary phase spend more time interacting with it, resulting in slower migration. Conversely, compounds with a higher affinity for the mobile phase move more quickly. Now coming to the next method of purification, that is membrane processes. Reverse osmosis and ultrafiltration utilize semi-permeable membranes to separate molecules of different sizes. Reverse osmosis is a separation process where the solvent molecules are forced by an applied pressure to flow through a semi-permeable membrane in the opposite direction to that directed by osmotic force, and hence is termed as reverse osmosis. In downstream processing, reverse osmosis can be employed to concentrate valuable components in liquid, such as proteins, enzymes, or pharmaceutical compounds by selectively removing water molecules. This concentration step can help reduce the volume of the solution, increase the product yield, and facilitate subsequent purification processes. On the other hand, ultrafiltration can be described as a process in which solutes of high molecular weight are retained within the solvent and low molecular weight solutes are forced under hydraulic pressure around 7 atmospheres through a membrane of a very fine pore size. By selectively allowing the passage of solvent and small molecules through the membrane while retaining larger molecules, ultrafiltration can efficiently concentrate biological products. This concentration step is essential for increasing the product yield, reducing the volume of the solution, and facilitating subsequent purification steps, just like reverse osmosis. Already discussed earlier while discussing filtration processes, permeate is that part of the feed that does pass through the membrane, and retentate is that part of the feed that does not pass through the membrane. Now the main difference between reverse osmosis and ultrafiltration lies in the size range of particles they can separate. Reverse osmosis uses a semi-permeable membrane with very small pores. On the other hand, ultrafiltration uses a membrane with comparatively larger pores, typically in the range of 1 to 100 nanometers. Due to their different separation capabilities, reverse osmosis and ultrafiltration find applications in different fields. Reverse osmosis is commonly used for desalination of seawater and brackish water, water purification, and the removal of dissolved solids and contaminants. Ultrafiltration is utilized in the purification and concentration of proteins, enzymes, polysaccharides, colloids, and particles. Other purification methods include use of liquid membranes. A liquid membrane is literally a membrane made of insoluble liquid. Liquid membranes involve the selective transport of solute across a liquid phase, acting as a barrier between two immiscible phases. It consists of a liquid phase, an organic solvent, or a thin oil film selective for a given solute, and serves as a membrane barrier between two other phases of aqueous solution or gas mixtures. Extraction takes place by the transfer of solute from one liquid to the other. They are of great interest in the extraction and purification of biologicals for the following reason. First, large area of extraction, then separation and concentration are achieved in one step. Scale up is relatively easy and it is a highly selective process. Its ability to selectively extract, separate and concentrate specific compounds make it a valuable tool in downstream processing for the purification and recovery of target compounds from liquid solutions. Liquid membranes are commonly used for separation and extraction processes in various industries such as chemical processing, pharmaceuticals, and environmental remediation. They are particularly useful for the extraction and recovery of metal ions, organic compounds, and pollutants from aqueous solutions. The separation process in liquid membranes occurs through a series of steps. First is the contacting phase. The liquid solution containing the solute to be separated is brought into the contact with the liquid membrane. The solute of interest selectively dissolve into the liquid membrane phase based on their affinity and partition coefficients. Second stage is diffusion and transport. Once the solutes are dissolved in the liquid membrane, they diffuse through the membrane phase. This transport process can occur by simple diffusion or facilitated transport mechanisms depending on the solute and membrane properties. Third step is the stripping phase. The solutes that have diffused through the liquid membrane phase are then transported into a receiving phase, often referred to as the stripping phase. The receiving phase is typically an aqueous solution or another solvent that can dissolve the solute. Fourth step is the recovery. The solute is then recovered from the receiving phase using techniques such as precipitation, evaporation, etc. for further processing or analysis. Liquid membranes are further of three major types. First is the emulsion liquid membrane. An emulsion liquid membrane can be visualized as consisting of a bubble within a bubble. So here R is denoting the receiving or product containing phase, F is denoting the feed or the source phase, and E is designating the membrane. In emulsion liquid membrane, the innermost bubble is the receiving phase, the outer bubble is the separation skin or membrane, anything that is present outside the bubble is the source phase. It involves the use of an emulsion, which is a dispersion of immiscible liquid phases where one of the phases acts as a liquid membrane. Second type is the supported or immobilized liquid membrane. Liquid impregnated or immobilized in the pores of a thin microporous solid support is defined as a solid liquid membrane. This immobilization provides structural stability to the liquid membrane, allowing for easier handling, separation, and reuse. Common support materials used in immobilized liquid membranes includes porous ceramics, polymers, glass fibers, etc. Third is the bulk liquid membrane, which consists of a bulk aqueous feed and receiving phases separated by a bulk organic water immiscible liquid phase. The liquid membrane phase is typically immobilized in a porous support material such as a hollow fiber or a flat sheet, which acts as a physical barrier to prevent the mixing of a membrane phase with the other liquid phases. Coming to the last process of purification stage, that is crystallization. Crystallization can be defined as the solidification of a liquid substance into a highly structured solid whose atoms or molecules are placed in a well-defined three-dimensional crystal lattice. The smallest individual part of a crystal is called a unit cell. The crystal is made up of millions of such unit cells. Crystallization is primarily employed as a separation technique in order to obtain pure crystals of the desired substance from an impure mixture. The first step in the crystallization process is to find an ideal solvent. The desired compound should be very soluble in the solvent where it is hot near the boiling, but only slightly soluble at room temperature, and insoluble at even lower temperature. At the same time, the dissolved impurities, that is byproducts and unreacted starting material, should be highly soluble in the solvent at both high and low solvent temperatures. The compounds must precipitate out of the solution at cooler rates and is filtered to allow its separation from the solvent and the dissolved impurities. Crystallization leads to a high quality products with desired characteristics and specifications. The next slide shows the crystallization process steps. Crystallization is possible for both impure liquids and solids. They are dissolved in the ideal solvent to form a solution. So step 1 is the solution is heated in an open container, as you can see in the diagram. In step 2, the solvent molecules start evaporating, leaving behind the solutes. In step 3, when the solution cools, crystals of solutes start accumulating on the surface of the solution. In step 4, crystals are collected and dried as per the product requirement. The undissolved solids in the liquids are separated by process of filtration. The size of crystals formed during the process depends on the cooling rate. A large number of tiny crystals are formed if the solution is cooled at a fast rate. Large crystals are formed at a slow cooling rates. Now here you also need to know what are nucleation agents. Nucleation agents, also known as seed crystals or crystal seeds, are substances or particles that are introduced into a crystallization process to initiate the formation of crystals. Examples include inert materials like glass beads, sands, or specific powders, organic molecules, salts, or metal complexes. Now let's discuss the advantages of crystallization. First is purification. Crystallization is employed to separate a desired compound or product from impurities, contaminants, or other components present in the solution or mixture. The process takes advantage of the differences in solubilities between the desired compound and impurities under specific conditions like temperature, solvent, concentration, etc. By inducing crystallization, the desired product forms well-defined crystals while the impurities remain in the solution or form separate, less pure crystals. Second is concentration. Crystallization is also used to concentrate a desired compound from a dilute solution. By manipulating the conditions like cooling or evaporation, the solubility of the compound can be reduced, causing it to precipitate as crystal. This allows for the removal of a significant portion of the solvent and the concentration of the desired compound in the remaining liquid. Particle size control. Crystallization can be employed to control the particle size of the desired compound by adjusting the cooling rate or the presence of nucleation agent. The rate of crystal growth can be controlled, resulting in desired particle sizes. This is particularly important in industries where specific particle sizes are required for product performance or formulation. Polymorph control. Crystallization can also be utilized to control the formation of different polymorphs, that is crystal structures of a compound. Different polymorphs may exhibit different physical properties such as solubility, stability, and bioavailability. By carefully monitoring the crystallization conditions, the desired polymorph can be selectively obtained, ensuring consistent product quality. Then recovery and recrystallization. Crystallization is often used for the recovery and recrystallization of compounds from a mother liquor or a crude mixture. By dissolving the crude compound in a suitable solvent and then inducing controlled crystallization, the compound can be obtained in a more purified form. So we have discussed the purification methods that includes chromatography, membrane processes, liquid membrane, and crystallization. We have also seen that processes like reverse osmosis, ultrafiltration, crystallization can serve as both concentration and purification steps. Now in the next video we'll discuss the last stage, that is finishing or drying.